Talk about monoclonal antibodies, which were used recently in the experimental Ebola treatment called Z-Mapp. So monoclonal just refers to the fact that any one antibody is the product of a single B cell that divided into a series of genetically identical clones, hence the term monoclonal. So scientists will take a laboratory animal like a mouse, rabbits, or guinea pigs, and they will infect them with the target pathogen, or with a lab-adapted strain of the pathogen, which has similar antigenicity but has other advantages. Next step is to isolate spleen cells, which hopefully will have a series of antibodies that have a range of binding affinities for your pathogen. And then, in order to get them to grow in cell culture, because spleen cells are mortal, they die, you have to fuse them with a cancerous B cell called a myeloma cell. These myeloma cells have a very convenient type of cancer for us that allow them to grow indefinitely in cell culture. Also, they lack the ability to make their own antibodies. So when you fuse them together, you know that the hybridoma's antibodies will have originated from the spleen cells and not from your myeloma cells, which is important because we want the ones from the spleen cells. Finally, the myeloma cells lack the salvage DNA synthesis pathway. So when you incubate them in the hat medium, which blocks the de novo DNA synthesis pathway, the myeloma cells that haven't fused with your spleen cells will die because they lack the salvage pathway. Only the fused cells that have the salvage pathway genes from the spleen cells will survive. And you know that the spleen cells by themselves won't survive because they're mortal. Finally, you'll have a series of hybridomas which have different antibodies, and you select the ones with the best binding affinities using techniques like ELISA or Western blot. So the result is that you have an animal antibody, and this can be rejected by a human immune system. So you need it to be humanized. You will extract the DNA from the clone that you have, and you can go in and edit this constant region, which is the region that usually differs, and a couple of areas in the variable region to humanize the antibody. So the last step is to make sure that your antibody has a human type glycosylation. Glycosylation is the last step in protein synthesis. Since it affects the very outside of the protein, it can have a dramatic effect on binding affinity. So you want to make sure that your protein expression system will have a similar glycosylation as your human or as the animal in which you grew the antibody. So you know the binding specificity will be just as good. Thanks so much, and I can go on for 3 more minutes.